Hello everyone, I am Mong Jalan from the Urology Department of the First Affiliated Hospital of Anhui Medical University. Today I would like to share with you our research about molecular classification of clear cell renal cell carcinoma that recently published in iMeta. Kidney cancer is the 9th most common cancer all over the world, while renal cell carcinoma accounts for 90%. Traditional molecular typing and biomarkers only consider one type of data. Ignore the interaction between molecular changes of different omics. In this study, we collected multiomics data such as mRNA, link RNA, gene mutations, copy number alterations, and DNA methylation profiles. Molecular subtypes were revealed via multiple clustering algorithms with our package MOVICS. Let's take a look at the research results. Molecular subtypes were identified based on 225 patients from ccRCC cohort. 10 clustering algorithms were used to classify patients into 3 subtypes and perform further consensus. Results show that MOS2 has the best prognosis, and MOS1 has the worst prognosis. For the pathways, observed that MOS1 patients contain the activation of membrane protein targeting and acute inflammatory. As for MOS2, endothelium development, fatty acid catabolic, mRNA processing pathways were enriched. The activation of immune-associated pathways enriched in MOS3 significantly. In addition, there was a high immune cell infiltration in both MOS1 and MOS3, but MOS1 also showed the activation of immune-exhausted signature. Based on the submap algorithm, we found that patients in MOS3 can benefit from anti-PD-1 immunotherapy. We collected 2 immunotherapy cohorts for validation, and observed that 71.4% patients in MOS3 from the Checkmate cohort, 80%. MOS3 patients from Meow cohort achieved clinical benefit. We also analyzed the different genetic mutations. SETD2 mutations mainly observed in MOS1 and MOS3 and are associated with poor prognosis. SETD2 mutations result in reduced mRNA expression. SICTD2 activates the immune pathway of antigen processing and presentation, cytokine-cytokine receptor interaction. SETD expression is positively correlated with PLXNA2 expression. To further verify our findings, we performed immunohistochemical staining in 60 clinical samples and found a significant positive correlation. After knocking down SETD2 using lentivirus, immunoblotting showed that the expression of PLXNA2 was also significantly reduced. At the same time, knockdown of SETD2 promoted the proliferation, migration, and invasion. For the chemotherapy, we found that the MOS2 subtype was more sensitive to sunitinib treatment, which further validated with MTAB-3267 cohort. In addition, we also selected axitinib, a drug commonly used in clinical, and found that MOS1 patients had a significant response to it. In addition, SETD2 mutant cells were sensitive to PI3K/AKT signaling pathway inhibitors. To further confirm the stability of the MOS classification system, we screened 100 specific markers for each subtype and to reproduce the MOS classification in external cohorts. In GSE 22541, patients in MOS2 had the best prognosis, while patients in MOS1 had the worst clinical outcome. In GSE 40435 and GSE 53757, patients in MOS2 were mostly in early tumor stages, while MOS1 contained the majority of stage 4 patients. In summary, we identified 3 molecular subtypes of clear cell renal cell carcinoma with significant different prognosis based on multiomics data and validated them in external cohorts. We found that MOS1 is an immune-exhausted subtype, which can benefit from PI3K/AKT inhibitors. MOS2 is an immune-cold subtype, but is more suitable for sunitinib therapy. MOS3 is an immune-hot subtype, which can benefit from anti-PD-1 immunotherapy. SETD2 is a tumor suppressor in ccRCC, and knockdown of SETD2 leads to the promotion of cell proliferation, migration, and invasion.